Okay, many thanks for this wonderful opportunity to share this challenging, maybe, and of course fascinating insights on epigenetics with you. My name is Tomi Aalto. I come from Finland. I have a very diverse and interesting job history and background. I've been working, for example, in a company, German company called Festo in Stuttgart, Esslingen. In Finland, we have a Festo Direct and other sectors. I worked for Festo for 2.5 years, and so I've been working in automation companies, programming, making programming, robotic specialist, etc. But I'm also a teacher. Currently, I'm classroom teacher. So I have a master's degree in educational sciences in University of Turku, Finland. Year might be 1989 when I had my degree. So let's start this presentation by focusing on a couple of words and terms we can find and widely used in Genesis. And these words and terms, first of them is the verb bara. It's a Hebrew word, and basic meaning is to create. But how is it used? This verb, it occurs 48 times in the Old Testament, and in the whole Bible, this verb is used about 243 times in different forms. So it's quite a common verb. The subject is always God. It's never used of human activity, such as making something, building, or crafting. And we have to be very careful when we investigate this verb. It's not an allegorical verb. It has a very specific meaning and purpose. The word doesn't always strictly mean creation out of nothing. So we may have heard the term ex nihilo. But it always refers to a new, unique, original, and supernatural act of God. And interesting is that the context determines whether it refers to ex nihilo or to creating something new in order of function. And here are a couple of examples how it is used. Genesis 1:1, in the beginning God created, then the verb bara is used, the heavens and the earth. And it's also used outside of the Genesis creation account in several other books, like Psalms, create in me pure heart, O God. The key idea is that bara emphasizes that God is the sole, original and unique and the only source of all creation. So it's a very person way and person type how this verb is used. So it's dependent in a person, Elohim person. Another word is a noun, the noun min, which has a basic meaning, a kind, category or type or a group or something like that. And it is also widely used in the creation account in several verses. So, and it has repeated in expressions like according to its kind. So this noun helps us to understand what a kind means, what a min means. It has semantic content. It indicates that God created plants and animals with diversity, but also with some kind of boundaries or barriers, so that each kind reproduces according to its own kind. The term is not identical with the modern biological term species. It doesn't mean that. it describes a broader reproductive grouping that maintains order within God's creation plan. And it also has its own context. The term emphasizes that created organisms don't cross into other categories, so a dog doesn't become a cat or a duck doesn't become a sparrow, for example, or vice versa. But they remain with their God-given boundaries according to their kind or kinds. And so the key idea is that the noun min expresses both the diversity but also the limits and the order that God has established in creation. And now I ask you to remember these, especially these limits and boundaries and barriers, because I'm coming back to these terms during my presentation. Yesterday I gave a presentation about epigenetics, and in this presentation I have a lot of same slides, so I think there's a lot of repetition, but I think it's not a bad idea to repeat some important things and understand how things work at genetic and epigenetic and molecular biological level. So let's start with a very known example of so-called evolution. This is, I think everyone of us knows this example. So Darwin's finches, this is his idea that there is natural selection and some kind of random changes. So according to Darwin, evolution and natural selection work as follows. Some finches develop strong and thick beaks as a result of random mutations, leading them to become seed eaters. Finches with slender beaks become insect eaters, and thus natural selection directs birds with different beak types to acquire more food, giving them a competitive advantage. They produce more offspring than birds that obtain less food, thereby reinforcing the trait in subsequent generations. This results in differentiation, which leads to speciation. And this is the foundation of the entire evolutionary theory. But today we know, and many even secular scientists know, that Darwin was seriously wrong, because this theory, it doesn't work anymore. It has never worked, but it was only a theory. But okay, Darwin makes some conclusions, but today we know that his conclusions were absolutely wrong. And next I'm going to explain this, why he was so wrong. This is a study, about 8 years old study, picked up from the Forbes article. And these statements are written by scientists. They write that these species of finch have distinct diets which could explain the differences in methylation patterns, as diet is known to influence epigenetics. They also know that those birds are genetically pretty similar, almost similar. DNA is almost the same. And they also know that DNA methylation can be a source of heritable phenotypic variation. And they also know that environmental stressors, such as pollutants, diet, etc., different environmental factors, can change these methylation patterns. And for them, this is it's a new mechanism. And yes, epigenetic mechanisms have been known for about 20 years. In theory, the idea has been known about 50 years, but through a true scientific level, the most important mechanisms have been known only for 5 years. So, but let's continue. They also know that the field of epigenetics is just getting started. So this is something new, and that is what we are going to study right now. Today we know that the size and shape of finches' beaks vary according to the type of food. So if a finch starts to eat seeds, then its offspring will have stronger beaks, and if it begins to eat insects, then after just a few generations, the offspring will have thinner and more delicate beaks. We know that these rapid changes are not based on random mutations or selection. That's absolutely an outdated theory. So evolution believers, they have to find another theory by which they try to defend their ideological foundation. Another excellent example of epigenetic regulation is the blind cavefish. Its widely has been used as evidence for evolution for decades, but is this based on random mutations and selection? No. But we still can find several articles on the internet which claim that over millions and millions of years these kind of changes happened and fish lost their eyes and probably the eyes don't come back and so on. But what happens in reality? The eyes are restored after the fish return to a light environment. And it doesn't take millions of years, only a couple of generations. So it happens very quickly. So there is some kind of epigenetic switch that turns to another position when fish live in a dark environment. So this sounds very designed, doesn't it? Because what does a fish, what does it do with eyes in a dark environment? It doesn't make sense to hold eyes in a dark environment. So it's best to leave them without growing. So this exactly points to designed features. But the scientists were shocked when they realized that the eyes are restored after fish returned to a light environment, and especially that it doesn't require millions of years, it doesn't require thousands of years. This happens very quickly, after 2 or 3 generations only. Italian wall lizard is also a very famous example and so-called evidence for the evolution believers, but today we know that this rapid change also was based on changed diet, nothing to do with random mutations or selection. Here's the summary of the study. Scientists were almost shocked to discover how quickly the lizards have undergone radical changes. Their behavior had changed, their jaws had lengthened, and most surprisingly, their intestines had developed a ring-shaped muscle called the cecal valve. All of this had happened in just a few decades. The reason was clear. The lizards had to switch their diet from insects to plants. The scientists didn't find any consistent changes in the lizard's DNA. But as we can see, this was the new structure in their intestines. And how long did it take to grow this structure? What do you think? 30 years? 1 year? 4 million years. Yes, they say that. But in reality, it took only 1 generation. I've read a study that said that a lizard can grow the measure of its intestine in just 40 days. Just 40 days, 17% more of its length in 40 days. So these kind of changes, they occur very, very rapidly. And of course, evolutionary believers are shocked. They don't want to talk about these changes. I have few slides that I can almost skip very quickly because this presentation is quite massive. I have 65 slides, so let's go this through very quickly. But a bee larva grows into a queen because it's been fed royal jelly. The DNA is identical. Note. The ant grows into a soldier ant. Again, DNA is identical. No changes in DNA. Stick insect, they can grow wings, but nothing to do with changes in DNA, nothing to do with so-called mutations. Mammoths, different phenotypes, but again, genetic similarity is astonishing, based on epigenetic switching. Nautiloids, very different outcomes, but again, DNA is identical. These changes are based on epigenetic regulation. Butterfly metamorphosis, 4 different stages, but again, DNA is the same in all these 4 different stages. So it's about epigenetic control of gene expression. Human skin color, an example from 2016 from Great Britain. Amelie and Jasmine are monozygotic twins, means that their DNA is absolutely identical, it's the same, but they have a different skin color. So we can conclude that skin color is not determined by the DNA. It is controlled by epigenetic mechanisms such as alternative splicing. I'll come to that later. Different plants, these are variations of the wild mustard plant. So different sprouts, cabbage, kale, Brussels sprouts, broccoli. Virtually it's the same plant, different variations. DNA doesn't change. Or if it changes, then there are no explanatory changes that could explain these changes within these different species or different variations. Different birds. Wie können 9 Arten so unterschiedlich aussehen und dennoch genetisch nahezu identisch sein? Was that correct? Yeah. Okay, thank you. You can go on in German. Maybe next time, yes, thank you. So, because I'm a teacher, I would like to provide with the most important question for a middle school or a high school biology teacher. And it's based on this fact. Every one of us in our bodies has about 300 different cell types. For example, skin cells, liver cells, etc. So there are only a couple of exceptions. All of our cells contain exactly the same DNA sequences, meaning that their genome is identical, of course, because we have started our life from one single cell that has started to divide to 2 and 4 and 8 and 16 and 32 and 64 and so on. So when the cell is copied and replicated, the DNA is, of course, also copied. That's why in every cell in our body is the same DNA. There are only a couple of exceptions. There are neurons in our brain. It's remarkable that in every cell in our brain there is a unique DNA sequence. It's incredible. Also, T cells used by our immune system, they might have different DNA sequences, but all the other cell types, they have exactly the same DNA. So now we can make the question, why does a skin cell have a different identity and function than a muscle cell, even though their DNA is exactly the same? So this is a basic question. It's a very logical question. But is this question asked for pupils and students in schools today? No. Why? Because evolutionary believers and supporters, they don't want to teach our students how the cell differentiation happens. Because if this was taught in our students, for our students, I think that most of our students wouldn't believe in evolutionary theory anymore. But this is not taught to students. So we can conclude that DNA does not determine the cell's function. And we can continue asking then what does determine the cell's function? And now we are coming to epigenetic regulation. But before that, I ask you to read this very important statement. I enjoy this glass of water. This is only 6 years old, but it's very important. Scientists have concluded that the nuclear genome has lost its function as the unique and exclusive determinant of inherited characteristics due to mounting waves of data revealing the complex epigenetic networks that heritably control genome expression. So they know that the DNA doesn't control anything. There is another information layer called epigenetic information layer that controls how DNA is read and so on. And because we have 300 different cell types in our bodies, we know that there has to be some kind of differentiation or some kind of mechanism that causes these differentiation happen in our cells. So we have to ask why cells are different even though their DNA is same. We can have muscle cells, blood cells, all kind of different cells, but the DNA is the same. How is this done at the cellular level? Let's have a closer look. The word epigenetic, it consists of two words. Epi is a Greek word meaning on top of or outside of something. That means outside information that is outside of DNA. And that mostly means some kind of chemical layers of markers. For example, CH3 molecule, which is a hydrocarbon group, a methyl group, and this is called methylation. And they have a very important meaning and purpose. They direct the reading of DNA, so they control how DNA is transcribed. It means how it is read. They also regulate several number of cellular other mechanisms. They can silence DNA and prevent it from being transcribed. So by methylation, the cell can, in a way, suppress or hide, keeping them outside of reading. And again, by using different methylation or without methylation, the structure can be open and readable and allowable for transcription. And it's good to know that about 80, in some cases more than 80% of plants' genomes are silenced. That means that plants have a great adaptation potential. And we know that without these epigenetic profiles, markers, the cell is an undifferentiated stem cell. It has lost its identity, its function, its functionality, and its job. It doesn't know what to do. So we know that the cell needs epigenetic programming in order to perform its designated function. And also important to know that methylation stabilizes the genome. This is important information because viruses, they don't have their own methylation production. So they kind of steal the methylation from their host cells. And that means that the host cell loses its own methylation profiles. And it might result and lead to very serious diseases. Viruses are dangerous. Yes, they need methylation in their own gene expression, yes. But they don't have, they don't have their own methylation production mechanism. Yes, yeah. Mm. And they steal the methylation from you and me, from ourselves. What do you mean?
It's always the virus would copy the whole string. It's not right. It's not correct that he had to copy the whole string.
Whole string of DNA.
Yes.
Their own. Yeah, they do. Yes. Yes, they have to do that because they have to copy themselves. Yes. But they copy, but they also read some sections of their own DNA. They don't read the whole section. Depends on virus type. Yes, yes. There are three main types of epigenetic information. The most common is the DNA methylation, and then we can have a look soon at histone epigenetic markers. They are very complex. And the third group is RNA signaling molecules, and mostly we are talking about non-coding RNAs. Here's the picture of DNA methylation, and we know that in eukaryotes the methyl group is attached to a cytosine base, mainly at so-called CPG sites, which means that at the DNA sequence the C base is followed by the G base, that is called a CPG site. The small p stands for phosphate. In prokaryotes, the methyl group is mostly attached to adenine base, so they have a different system. Maybe, I think this is something to do, of course, with design. You might have heard in school times that might be some kind of gene in a genome, and one sequence of DNA codes for one RNA molecule that further codes for one protein. Are you familiar with this kind of idea? Now I have to say that we have... Yes, we have to forget this outdated theory. It doesn't work this way. Because the cell can produce thousands of different proteins from the same DNA sequence without changing the DNA. How is it done? Let's have a closer look. For example, we humans, our cells they only have about 20,000 DNA sequences used and read for protein production. It means it's less than an earthworm, for example, nematode, which has 22,000 protein coding genes. We have only 20,000 so-called protein coding genes, but the number of different proteins in our bodies might exceed 6 million. So you can make a short calculation that how many proteins on average are produced by reading only one DNA sequence. The answer is about 300. 300 different proteins are produced by reading only one DNA sequence. Incredible mechanism, isn't it? Das ist sehr gut. Yes. This is based on a very complex mechanism called alternative splicing, and it has epigenetic control and regulation by DNA methylation profiles, histone epigenetic markers, and non-coding RNA molecules. But this also makes us ask a question: what is a gene? Because actually there are not some kind of genetic units in the genome, informational units. There are no units. There are no genes in the genome. The gene actually is somewhere outside of the genome. I would say that the gene can be found at the RNA level. But at DNA level, we cannot find genes. There might be a very little share, a little minority, perhaps 1% something of our genome is directly encoded to RNA and further to a protein or other RNA molecule. So this picture might be quite difficult to understand, but I try to ravel it up. This illustrates how alternative splicing works. Please focus your attention on those colored segments. They are called exons, and the cell's mechanisms have selected them from different locations in the genome, even from different chromosomes. These sequences are joined together and transcribed into RNA molecule. The cell can modify this so-called pre-mRNA and in different ways by combining useful exons and removing unnecessary introns. In this way, the cell can create various combinations from the transcribed sequences and further produce different proteins. No modifications to the DNA are needed. This mechanism is epigenetically directed and regulated. And in this example, we have only 3 different proteins, but they are all of them are coded by reading only one DNA sequence. The maximum amount of different proteins based on only one DNA sequence has been found in Drosophila melanogaster, which means the fruit fly. Yes, and the number is 38,016 different proteins by reading only one DNA sequence. More than 38,000 different proteins. Sounds designed. So here it is, an example. It's the insect gene DS-CAM, encodes more than 38,000. So we can now understand why organisms have so remarkable adaptation potential without changing DNA. So now we have learned that there are no single information units called genes in genomes. That's why most of scientists and researchers, they want to move the gene concept from the DNA level to the RNA level. It might be more sensible, and yes. We also can conclude that DNA actually is passive information data repository. It means that the cell picks useful DNA sections because it has a mechanism, but it is using microRNAs and other non-coding RNA molecules to find correct DNA sequences from the genome, and it picks and transcribes and joins them together to make a pre-mRNA molecule that is further possibly modified also. I'm not the only one who has concluded that DNA is passive information. Sorry. So Dennis Noble, you may have heard of him. He said that the modern synthesis has got causality of biology wrong. The DNA on its own does absolutely nothing until activated by the rest of the system. DNA is not a cause in an active sense. I think it's better described as passive database, which is used by the organism to enable it to make the proteins that it requires. And also other statements of this idea that DNA is actually passive information. It doesn't determine organism traits or characteristics. This is very interesting. You might have now learned something about alternative splicing, how it works. But did you know that after a loss of information due to harmful mutations, genetic errors, the cell is able to compensate for the loss of information by programming the alternative splicing machinery to function in a more efficient and complex way? That's incredible. So genetic degradation is a biological fact. It happens all over the world in all kinds of organisms. But the cell still has a function and functionality to compensate for the loss of information. So this is astonishing for me, incredible. And here are a few examples of how organisms are using alternative splicing mechanism. For example, the moth called Macrosilix maya, you can see that it has a great ability to paint pictures of flies on its wings. And it seems that those flies are eating something birds poop, yes. So it's a lot to do with the camouflage. And on the left side, on top of, there's a small butterfly called Goniurellia tridens. How is it pronounced in Latin, maybe? And other examples. These all are based on alternative splicing. Nothing to do with random mutations and selection. Also, these so-called adaptive radiation, very rapid changes within these cichlids in the lakes of East African lakes. These are also based on alternative splicing. No changes in DNA are needed. Scientists may have found some changes in DNA, but those changes, they don't explain these rapid changes within those fish. Okay, let's get deeper. Do we have time? Some. Did you know that every genome of every organism is very, very compressed and packed information? It is done by using nucleosomes and histones, which are like ring or circle-shaped structures that DNA is coiled around. We can see a couple of pictures that help us understand how this compression mechanism works. So when we open up the chromosome structure, we see a chromatin structure, and again we see chromatin fiber, and we can see those circle-shaped structures called nucleosomes. And again, if we open that more, then we can see DNA strands. So here's another picture of this structure. And scientists use terms such as heterochromatin, which means closed chromatin. That means that it's some kind of suppressed information. It's hide it. The cell doesn't want to read it. It's silenced information. And when the DNA is transcribed, the structure needs to be opened. So that kind of structure is called euchromatin, and it's loosely packed, and that might open the structure, and transcription can happen. And please focus on those small tails, on those sector-shaped structures called histones. Those tails have a very, very important purpose for gene expression and many, many other mechanisms and information handling systems. Those epigenetic marks on those histone tails, these are known to be at least 20 different types today. And they have many control and regulatory functions, such as they regulate alternative splicing, they determine how the methyl groups are located on top of DNA, they affect post-transcriptional modification and also post-translational modifications. So several important jobs and function tasks. Histone epigenetic marks function like a biological memory registry database. They synchronize and coordinate multiple layers of information to work together and in a timely manner. And we can think that and ask, are those histone marks inheritable? Yes, yes they are. And I will soon show you how it's done. There is a very, very interesting mechanism how these are inherited. So here we have a larger picture of histones. The whole structure is a nucleosome, and these 4 different parts are called histones. And from every histone, in every histone there is a histone tail that may carry even hundreds of different kind of epigenetic marks. Those are mostly amino acids. It consists of from 4 to 9 atoms. So we are at atom level right now. So it's very difficult to analyze and study and make pictures of these, because the technology doesn't allow to go so deep on the atom level, at the atom level. But what is interesting is that these histone marks, and especially their combinations, they affect in many ways how the cell uses DNA and how it modifies RNA molecules, etc. They have very, very many tasks and functionalities. And scientists have to, they try to solve the histone code. It is called histone code. Today they have solved about 1%, perhaps not even that. The code is so complex, it's very complex. And because histone markers, it's information about information that might be information of another layer of information. So it's information about information about information about information. which means metadata of metadata. Evolution, of course. Sounds very designed, yes. So today, okay, there are 12 different market types, but I should update the number. It's almost 20 or even more than 20 different market types today. Very complex code, heritable information, and this is what epigenetic memory is based on. And Mr. Darwin also had some kind of idea of heritable memory. Yes, gemmules, yes. And he talked about the fitness, and it has something to do with memory. But today we know that this memory is based on epigenetic markers that are heritable. Then we can have a closer look at RNA signaling molecules. There are different types of these RNA molecules, and we should know that there are more than 200 different epigenetic marker types known to attach to RNA molecules. At DNA level, we only know about 4 or 5 different information marker types that can attach to DNA. But at the RNA level, there are more than 200 different marker types. And this is also some kind of biological code. It's a programming code, and scientists try to solve it. But again, the mission, yes. Yes, they have a lot of tasks, depending on their job. But if they are signaling molecules, they transmit signals from the, for example, environment and environmental factors. And by carrying different epigenetic markers, they carry the signal into the cell, and further they can bring the information to those histones and histone tails. And of course, they affect many biological pathways. Yes, but a lot of functionality. They also regulate cellular differentiation programming. They regulate tissue type, organ function, and even body plan. So the outcome, it doesn't come from the DNA. It comes from epigenetic regulation. And this slide is very important to understand. So let's have a look at this slide and stay with this a little more time. The direction and transfer chain of biological information when we're talking about adaptation of organisms. So there are some kind of environmental factors, and then the organism senses and receptors. They monitor environmental factors, and further receptors or the brain transmits this information to cells through signaling pathways using various signaling molecules. And again, the cell's epigenome responds to these signals, and the cell's epigenetic program changes. And after that, the function of the cell, tissues, and organs adapt to changes in the environment, and the changes are inherited by offspring. So any change is based on complex mechanisms, nothing to do with random mutations or selection. This slide might better answer to a question. For example, long non-coding RNAs, they have these kind of extremely complex and important tasks. They can regulate and control cell differentiation programming, direct and regulate formation of healthy tissue type, mediate and control organs to work correctly, regulate and control body plan, etc. And when evolution believers often want to compare human and chimp genomes and DNA. Okay, today we know that even at the DNA level, the similarities are about 86%. But at the long non-coding RNA level, that are very important in terms of cell differentiation, so on, the similarities are about 10% only. That's mainly because of we humans have more than 9 times more of these complex long non-coding RNAs than chimps have. And we should know that these molecules, they don't tolerate mutations. Even one single mistake in one single RNA molecule might lead to serious diseases, such as cancer. There are several signaling pathways in our cells, and I will not stay with this slide a long time because we don't have time to do that. You can find this information by reading the book I've authored 2-3 years ago, and it's translated into English this year. You can find this from Finnish Creation Society, luominen.fi, and the other book I've written together with Dr. Pekka Reinikainen in Finland, Game Over Darwin. This book is based on a basketball game between a platypus and Darwin. And I can say that platypus wins the game by points 251 to 0. Yes. Can I find the picture? Yes, summing up. You can see something. Yeah. You can find this in Amazon. Game Over Darwin. 31 euros. Okay, epigenetic information is dynamic and reversible because there are specific enzymes that are responsible for writing information, reading information, and even erasing information. So there are very specific mechanisms doing all that, so that the adaptation is a very dynamic process. A rapid response is possible due to an efficiently functioning information layer called the epigenome, which is kind of response some kind of buffer that handles the information between the cell and the environment. It's called the epigenome. And it is based on epigenetic readers, writers, and erasers. And here's a picture illustrating how they're working. So there are writers writing those markers to those histone tails. There are erasers taking them out, and readers that can read what kind of markers are there on those tails. And we know that you may have heard of epigenetic switches, but actually they work like dimmer switches, like knobs. And this mechanism is based on three-dimensional shape of the chromatin. And if we add to that dimensionality, we add time aspect, then we will talk about four-dimensional genome. So it's called, okay, I'll come to that later. Epigenetic memory, I just talked about it a few minutes ago. There are different types of epigenetic memory in cells, and we can distinguish between them on how stable the epigenetic modifications are in terms of inheritance. There are different types of epigenetic memory. Some of them are more stable than other types of memory. But the most fascinating mechanism is extracellular vesicles that the cell uses for passing this epigenetic memory not only to neighboring cells, but the cells from ancestors to the offspring. This occurs during the physiology of reproduction. And in addition to the inheritance of DNA, non-coding RNA molecules transmit epigenetic information to the tails of histones in embryonic cells, which direct the setting of epigenetic profiles in the embryo and fetus. This is an incredible mechanism, and this is something we should understand. We know that the methylation profiles, those CH roots, they are usually weakly inherited, and they mostly are erased during the first stages of embryonic development. But there is much stronger and stable mechanisms to transmit epigenetic information across and over generations. So it means that traits and characteristics, adaptational states and so on, they are stored in the histone database. And again, non-coding RNAs, which are encapsulated in the small nano-sized bubbles called extracellular vesicles, they transmit this kind of complex information to the next generation in the physiology of reproduction. Before we look at that mechanism more accurate level, we should know that the cell has also function and ability to do intentional changes in DNA. So the theory of random mutations, now we know it's really outdated, it doesn't work anymore. If we talk about random mutation, then we can talk only about genetic mistakes and errors. But this is fascinating. The cell can make intentional changes in DNA, and of course RNA too, which can and do lead changes in phenotypes. A very recent discovery is the mechanism by which the cell can, by purpose, modify both RNA and DNA sequences. And it uses specific enzymes for doing this job. It uses ADAR and ApoBec and A3G enzymes. And when you read studies of this mechanism, they are quite new, and scientists are almost shocked after realizing that the cell has this kind of abilities. You can find articles by using keywords such as mutations are not random. Yeah. Yes, the environment factors might trigger the cell to make those intentional changes, and it's lots about adaptation, how the organism has to adapt to different environmental situations and factors. Yes. Speciation and species. Yes. Both, yes, yes. And at individual level, during our own lifetime, our cells can make intentional modifications that might affect protein production. Not direct examples right now, because this is also quite new for me, and I haven't found living examples yet, but I'm studying. Yeah. So, speciation is a man-made word, because today we know that most of adaptational events and so-called speciation events are based on both alternative splicing and also RNA editing. And the third one, which is an important source for phenotypic variation, is so-called genetic mistakes, errors. Because at some rate, the cell has to rearrange DNA after the loss of information. The cell has to rearrange DNA in order to have correct and enough those GC pairs compared to AT pairs. There has to be enough GC pairs in the genome in order for the cell to have enough information to construct RNA molecules and proteins, etc. So different mechanisms behind speciation, but none of them results in any kind of evolution. No. They cause variation, but nothing to do with assumed evolution. No information increase. And RNA A to I editing is a very, very complex and astonishing mechanism. I can tell you something about that a little later. So we know that so-called speciation might happen without a single change in the DNA sequence, and it also might happen very quickly, only after a couple of generations. We have some examples of that. Mostly speciation is based on epigenetic regulation, which causes also errors in DNA, and this results in that the cell rearranges the DNA, and after DNA rearrangement procedures we can observe different phenotypes, and then we can say that, okay, speciation has happened. But actually it's about variation, and we can compare it to dog breeding. So RNA editing is a quite complex and but also fascinating mechanism, especially A to I RNA editing, because A to I refers to the modification of adenosine into inosine. Inosine is not a base found in RNA. Rather, adenosine has been modified by an enzyme from the ADAR family, transforming it into inosine. When messenger RNA containing inosine goes through the translation process during protein production, the translator, which is the ribosome, interprets inosine as guanine, which is the fundamental base in RNA. Thus, the event is equivalent to an A to G change in DNA, but this is a temporary change and also reversible change, just like other RNA editing events. So this is not the only RNA editing mechanism the cell has. In fact, it can change any RNA base to another base. But this is the most common and maybe the most studied mechanism. But it's incredible because we humans and especially those who are behind this desk, our technicians, they know the network address translation system. You have heard about that. It's the same system. Yes. Because it's about masquerading, you know. Yes. So it appears to as guanine to the translator, and we humans use this kind of similar technique in network traffic through what is known as network address translation, and it's a masquerading technique. So the cell has ability to make all these changes at RNA level, from any base to another. Some of them are less studied and some more studied. And now we are coming to the last, but maybe also the most complex mechanisms of how these epigenetic information layers are inherited. And now, in the beginning of this presentation, I asked you to remember a few words that are mentioned in the Bible that describes how God created the kinds and how the barriers and boundaries between kinds are maintained. And now we are coming to that information. Those nano-sized bubbles called extracellular vesicles, such as they are called exosomes and microvesicles, they can carry and transport, for example, non-coding RNA molecules, also proteins and snippets of DNA too, so several types of information. They can transport them between cells and even across generations. How is it done? Let's see. These non-coding RNAs, they influence and affect recipient cells by modifying gene expression patterns and chromatin structure. For example, sperm-derived small RNAs can carry environmental epigenetic information influencing offspring development. And the transmission of epigenetic signals through extracellular vesicles provides a non-DNA based mechanism for adaptation and inheritance. And here's an interesting statement. It's picked up from a study made in 2024. Yeah, it's pretty new. So these extracellular vesicles actively modify DNA, RNA, and histone profiles in recipient cells by delivering RNA and protein cargoes for downstream epigenetic enzyme regulation. So here's a picture of a vesicle, microvesicle. It's a nano-sized bubble. It has its own membrane. And we can see structures called tetraspanins and integrins, also lectins, ligands, receptors. What are these? What is this purpose? They act like keys, and they only fit to certain locks. So the keys has to fit with a certain lock in order to release its cargo. So the cargo of the extracellular vesicle can enter the recipient cell only if the key lock pair is compatible. And this is it's not only one, but maybe the last and strongest barrier and boundary mechanism that ensures that kinds are separated and distinct from each other. Because dogs vesicles carried in its semen fluid. Its vesicles are not compatible with cat's cells. So the vesicles, I mean the keys of the vesicle of dog, they don't match. They are not compatible with the cat's cells, which has its own locks. So they have to be compatible in order for the cargo to be released for the recipient cell. So we now have a very complex and incredible molecular level key lock mechanism behind biblical kinds. And now we have a better understanding of why barriers between different kinds are maintained and how they are maintained. There are, of course, other mechanisms, but this is one of the most fascinating mechanisms. So, key lock mechanisms are incompatible across different kinds. And interesting is also that when the cargo is released from the vesicle, it doesn't mean that it releases all of its cargo, because it's key dependent. If the cell and the vesicle uses a certain key to a certain lock, it might release only certain RNA molecules, or certain proteins, or certain DNA snippets. So it's filtered information. Incredible mechanism. Unfortunately, epigenetic regulation results in genetic degradation. You can find information by using keywords, hotspot mutations, and it's about it's associated with the methylation of cytosine mostly. But because of the lack of time, I will not handle this question long, so let's go this very quickly right now. But the main result is that so-called GC content, that means GC pairs in DNA, it gradually shifts to AT content, and this leads to problems in the cell. The problems are a loss of genetic diversity, the cell's ability to produce a variety of proteins diminishes, and the immune system weakens. Eventually, the organism is under the threat of extinction. But there are certain profiles called CPG islands. It means that there are several CG pairs in a row, side by side, even thousands of them. The cell uses these kind of CPG islands for gene or regulation or transcription regulation. Means that where transcription starts and where it stops, the intensity and duration and strength of transcription. But due to one specific mechanism, when a cytosine base is methylated, it easily converts to thymine. And this results that to a GC pair easily turn to AT pair, especially if the cell is replicated. And we should know that the cell has no mechanism to rebuild these kind of CPG islands. So it means that these islands, they only collapse. There is no known mechanism by which the cell could rebuild CPG islands. Though this argument, a very strong argument against the theory of evolution. I have challenged several artificial intelligence applications such as ChatGPT or Gemini or other, and they have admitted that yes, there is no mechanism to build new CPG islands, so evolution has no mechanism. Genetic decay is an inevitable phenomenon. The cell tries to balance and correct this situation by copying more GC pairs by using, for example, transposable elements, but it cannot do it in such a way that it could repair the situation. It means that genetic degradation is an inevitable event in all kinds of organisms in the world. You can find by studying statistics in disgenet.com, you can find that there are more than 2 million genetics. They are called gene disease associations in the human genome worldwide at population level. More than 2 million genetic mistakes. However, the human chromosome number is not reduced because of some certain long non-coding RNA molecules and proteins called proteins. We humans, we don't speciate because the Bible says we are created in God's image. Yes, we don't speciate. And today we know the mechanism why we don't speciate. A few words about mutational load. This is something that every serious scientist and researcher knows. They know that if much more than 3% of genome is functional, evolution becomes a destructive process. And the percentage might change. Someone has said that the percent is 10% or something. Whatever, today we know that more than 90% of the human genome is to be transcribed. And for the rest of the genome, scientists have found several regulatory functions and functionalities. So the fact is that mutational load remains, but there is no buffering junk DNA layer. So what are our conclusions? Information loss is easily observed by analyzing, for example, number of chromosomes within diploid organisms such as mammals. This example is within canids. We can see wolves still have 78 chromosomes, but after 7 speciation steps, red foxes only have 34 chromosomes. A very rapid loss of information. And here are the other examples: horses, muntjacs, mice, giraffes, and so on. So genetic entropy is an inevitable phenomenon. The best example is the famous Lenski's experiment. Did you know that after about 90,000 generations of bacteria, they have lost about 3% of their DNA due to genetic errors? That experiment started in 1988, but after 90,000 generations only. So We should also know that deletions are 4 times more common than any type of insertions. This is also a very strong argument against the theory of evolution. So, we are now close to make the conclusions. Epigenetic regulations or a loss of information. This is what any change in organisms is based on. Nothing to do with assumed evolution or natural selection. So, our conclusions are that true science, serious science, supports the biblical creation account. And this is what the change is based on. Organismal variation within created kinds occurs epigenetically through the switching of epigenetic toggles and sliders and knobs, which are based on epigenetic readers, writers, and erasers, and so on. Random mutations are not the cause of the organismal variation or adaptation. And these changes, they happen very rapidly, very fast. So we can conclude that any change in organisms is based on pre-existing biological information or a loss of information that results in a rearrangement of DNA, rearrangement of information. So there is no mechanism for evolution. All organisms are genetically deteriorating. We can observe a rapid decline in chromosome numbers, especially in diploid organisms. And as I said, there are more than 2 million genetic errors in the human genome. However, no human speciation. The number of fully random or fully beneficial mutations in human genome is 0. There are no beneficial mutations in the human genome. And we can ask, why would one of the most important questions, the question of the origin of life and humanity, be written metaphorically? So based on scientific observations, the biblical creation account is based on true events. Creation is mentioned over 200 times in the Bible, evolution never. The truth can be found in the Bible. So at last I want to share these verses with you. But ask now the beasts, and they shall teach thee, and the fowls of the air, and they shall tell thee. Or speak to the earth, and it shall teach thee, and the fishes of the sea shall declare unto thee. Who knoweth not in all these that the hand of the Lord hath wrought this, in whose hand is the soul of every living thing, and the breath of all mankind. So thank you very much. Vielen Dank. Gottes Segen.